Let's discuss MCQ number 33. Which statement concerning the defense in the body against infectious disease is not correct? So we have to pick up the statement which is not correct against, or which is not correct concerning the defense in the body against infectious disease. So it says antibodies against specific antigens are produced by plasma cells in passive immunity. This is wrong. So the first statement is not correct. The answer is A. Why the first statement is wrong? Because antibodies against specific antigens are produced by plasma cells in active immunity, not passive immunity. When antibodies are being produced, it's active immunity. When antibodies are acquired, it's passive immunity. So if antibodies are being produced in me, that is an example of active immunity. But if I'm acquiring antibodies somehow, for example, I'm acquiring antibodies in milk, or I'm acquiring antibodies by injection, that is passive immunity. But they are asking about, they have written about produced. So whenever antibodies are produced by plasma cells, it's active immunity. So A is wrong. Obviously, if A is wrong, rest of the statements are right. Okay, so we don't need to discuss these statements, or we can. A specific immune response involves activation of B lymphocytes and T lymphocytes following recognition of and binding to a specific antigen. This is true. Following invasion by microorganisms, natural active immunity can be gained by initiating an immune response. This is true again. Lysosomes fuse with vacuoles that have been formed by phagocytes, which contain invading microorganisms. This is true as well. Okay. Let's move on to MCQ number 32. During a primary immune response, the following events occur. In which order will the events occur? So, what is primary immune response? When the antigen enters the body for the first time. Always remember this point, that whenever antigen enters, and if we talk about lymphocytes, so which lymphocytes are the first to be activated? These are T helper lymphocytes. So T helper cells will identify the specific antigen when the antigen enters the body for the first time. So this is the first statement. This process comes first. This process happens first, that the T helper cells identify a specific antigen. When they identify a specific antigen, they are activated and they start secreting cytokines. So always remember this point, that T helper cells secrete cytokines. And T helper cells secrete cytokines in order to activate other cells of the immune system. Because as I mentioned earlier, that cytokines are chemical messenger molecules, right? Which go on and give messages to other white blood cells. And with the help of these messages, other white blood cells are activated. Okay. So, fifth is the first event. Fourth one is the second event. Then after T helper cells secrete cytokines, this will activate B lymphocytes. So B lymphocytes with specific cell surface receptors divide repeatedly by mitosis. So whenever B lymphocytes are activated, they divide by mitosis. So this is the process which will happen next. And when the B lymphocytes divide by mitosis, they increase in number, and then some of the B lymphocytes will specialize into plasma cells. So this is fourth one, and then the plasma cells will secrete antibodies. So this is the fifth one. So what is the sequence? 5, 4, 2, 1, and 3, right? The answer is C. So this is the order in which the events will occur. Let's move on to the next MCQ. Which type of immunity occurs as a result of ingestion of antibodies by an infant through its mother's milk? So it says ingestion of antibodies, right? So whenever a person is acquiring antibodies, it's passive immunity. Whenever a person is acquiring antibodies, it's passive immunity. If the person is acquiring antibodies by injection, it's artificial. If the person is acquiring antibodies naturally, for example, by ingestion of milk, that is natural. So the answer is D. This is natural passive immunity, right? And what is artificial passive immunity? When the person acquires antibodies by injection, that is artificial, right? Because in this case, the infant is receiving antibodies from the mother in breast milk, So because infant is receiving antibodies, it's passive immunity, and uh, ingestion of a milk is a natural process, so it's natural passive. The answer is D. MCQ number 30. A student wrote down 3 statements about antibodies. Which statements are true? Their structure depends on peptide, hydrogen, and disulfide bonds. This is true. This is definitely true. If you remember the structure of antibodies, antibodies are glycoprotein molecules. So the antibody has 4 polypeptide chains, and in each polypeptide chain, amino acids are joined together by peptide bonds. Then each polypeptide chain has a tertiary structure, so there are hydrogen bonds, and the 4 polypeptide chains are held together by disulfide bonds. So the antibody structure depends on peptide, hydrogen, and disulfide bonds. This is true. They are protein molecules with both tertiary and quaternary structure. This is true. Antibody, because antibody has more than 1 polypeptide chain, it has 4 polypeptide chains, right? So it has a quaternary structure, and each polypeptide chain in an antibody has a tertiary structure. 4 polypeptide chains provide 4 antigen binding sites. This is wrong. What you need to know is that 4 polypeptide chains provide 2 antigen binding sites, not 4. So for example, these are 2 polypeptide chains and these are other 2, right? So the 4 polypeptide chains altogether provide how many antigen binding sites? 2 antigen binding sites, right? So the answer is B. What are the functions of T lymphocytes during an immune response? This is a repeated question, right? So if you remember, there are 2 types of T lymphocytes, T helper lymphocytes and T killer lymphocytes. So T killer lymphocytes destroy infected body cells. Both T killer and T helper lymphocytes differentiate into memory cells, and T lymphocytes have nothing to do with secretion of antibodies. So answer is C, 1 and 2 only. Let's discuss MCQ number 28. The graph shows the primary and secondary responses of immune system to antigens. What are responses X and Y? So guys, you need to know this graph. If you look at the responses X and Y, so you need to know that Y is primary response. Y is primary response and X is secondary response. I'll let you know why. So what you need to know is that when the antigen or pathogen enters the body for the first time, there is primary response. And then when the pathogen or antigen enters the body for second time or third time or fourth time and so on, it will be secondary response, right? So if a particular pathogen, for example, chickenpox virus, for example, entered your body for the first time, there will be immune response, and that will be primary immune response. So what your body will do, that it will initiate immune response and the virus will be eventually cleared. So in your life, if chickenpox virus enters your body the next time, it could be the second time, third time, fourth time, and so on, it will always be secondary response. What you need to know is that secondary response is faster than primary response. So how do we know that? How can we see from the graph that X is faster than Y? On y-axis, you can see concentration of antibodies in the plasma, and on x-axis, you see weeks after first or second exposure to antigen. So you can see that in Y, it takes how many weeks? It takes 4.5 weeks for antibody to reach peak concentration in the plasma. So it's a slower process. Primary response is slow because the pathogen enters the body for the first time and the body does not have memory cells, right? But if the pathogen enters the body the second time, the body has already seen it before. So the body has memory cells and the immune response is way too fast. So we can see that in X, antibody concentration reaches peak within 1.5 weeks, right? And this is all because of memory cells, because memory cells are there, and memory cells, they respond very rapidly. They divide and specialize into plasma cells. Plasma cells secrete antibodies, right? So there are memory B lymphocytes. which cause this to happen in secondary response. So what is the answer? What are the responses X and Y? You need to know that Y is the primary response and X is the secondary response caused by cloning of B lymphocytes formed during the primary response. So B lymphocytes formed during the primary response are known as memory cells, right? B lymphocytes formed during the primary response are memory cells, and in secondary response there is rapid cloning of these memory cells. D is wrong because they are mentioning about T lymphocytes. So in reality, T lymphocytes are also active in secondary response in such a way that there are also memory T lymphocytes, but we won't mention memory T lymphocytes over here because they are talking about concentration of antibodies, right? And antibodies are secreted by B lymphocytes. So we are done with question number 28. Now we can move on to the next MCQ. What happens when people are injected with dead bacteria? So guys, dead bacteria are an example of antigens. Like on dead bacteria, there are antigens. Okay. So if you are injecting people with dead bacteria, you are simply giving a vaccine which is known as inactivated vaccine. Inactivated vaccine contains dead bacteria. And what will happen, because bacteria contains antigens, there will be active immunity. So B lymphocytes produce antibodies, this is true. B lymphocytes produce antigens, no. T lymphocytes produce antibodies, never. T lymphocytes produce antigens, this is not true. Let's move on to MCQ number 26. Which row correctly matches a function with B lymphocyte and T lymphocyte? May become plasma cells, this is true for B lymphocytes and false for T lymphocytes. So A is wrong, right? B lymphocytes may become B lymphocytes may become plasma cells. So what we can do over here is that I will just circle which is right. So may become plasma cells, T lymphocytes will not become plasma cells, this is true. But B lymphocytes can become plasma cells, so this is not true. So I'm not circling this one. I'm circling the option which is true, okay? So this is wrong. May secrete antibodies, both of these are wrong. So I will underline what is wrong and I will circle what is right, okay? So this is wrong. Provides a cell-mediated response. So cell-mediated response is provided by T lymphocytes but not by B lymphocytes. What is meant by cell-mediated response? Cell-mediated response is when the cell destroys the pathogen. And what is meant by humoral response? There are two types of responses in immunity. One is humoral response and one is cell-mediated response. So cell-mediated response is when the cell goes and destroys the pathogen. For example, T killer lymphocyte goes and destroys the infected cell. So T lymphocytes provide cell-mediated immunity, but B lymphocytes never go and destroy a cell. What B lymphocytes do, they secrete antibodies, they secrete chemicals. That is humoral response. So response or destruction of pathogen by the antibodies is called humoral response, and destruction of pathogens by cells is called cell-mediated response. So T lymphocytes are involved in cell-mediated response, whereas B lymphocytes are involved in humoral response. So this is true. And this is true as well. Like T lymphocytes are not involved in humoral response. So I've circled that. This is true. And B lymphocytes are involved in humoral response. So answer is D. C is wrong. Right? Because of these underlying things, these underlined things are wrong. That's why the option is wrong as well. Okay? Let's move on to MCQ number 25. Antibodies can act in a number of ways to protect the body from pathogenic bacteria. Which event will not occur following antigen-antibody binding? Okay, so for this you need to know that how antibodies function. Antibodies bind to antigens and they can destroy antigens by number of different ways. So antibodies can bind to a lot of bacteria at a single time, causing agglutination of bacteria to reduce their spread. So this is actually true. We have to select that is not true, or that does not occur. Okay, so what you need to know that what antibodies can do is that antibodies with multiple antigen binding sites, antibodies with multiple antigen binding sites can bind to many bacteria at one time, forming a lump. This is called agglutination, and this prevents spread of bacteria. So increased susceptibility to phagocytosis, this is true as well. Like if bacteria are bound to antibodies, these antibodies can bind to a phagocyte, right? And this can cause increased susceptibility of bacteria to phagocytosis. This is true as well. Neutralization of toxins to make them harmless. This is also done by antibodies. So antibodies, what antibodies can do, that antibodies can bind bacterial toxins. So for example, this is bacterial toxin, and antibody can bind to bacterial toxin, and antibodies can neutralize these toxins. When antibodies are able to neutralize bacterial toxins, the antibodies are known as antitoxin, right? So antitoxins are antibodies that can bind to bacterial toxins and neutralize them. So this is true as well, which means that D is not true. Secretion of histamine to produce an allergic reaction. This is not done by antigen antibody binding, right? Okay. MCQ number 24, which describes a T helper lymphocyte. So antigen invades, recognition and binding to specific cells of the immune system, true. Activation of specific cells, growth and division of a specific cell, synthesize antibody, not true for T helper lymphocyte. We are talking about T helper lymphocytes. So always remember this point, that T helper lymphocytes secrete cytokines. The role of T helper lymphocytes is to secrete cytokines, and these cytokines will go on and activate other cells of the immune system. So remain in circulation, releasing cytokines, this is true. Kill infected host cells, this is not true. Okay, the answer is C. So we are done with MCQ number 24. We will solve remaining MCQs in the next video. Thank you for watching. Allah Hafiz everyone.